They named the mission Janus, 9, after the Roman god of doorways, beginnings, endings, and the thresholds between. Fitting, though no one realized just how literal that symbolism would become. The spacecraft had left Earth 5 years prior, bound for Europa, Jupiter's icy moon, its objective simple in wording but monumental in ambition: drill through the frozen crust and retrieve samples from the subsurface ocean rumored to exist beneath. No one expected success, not really. The mission was more performance than practicality, an expensive gesture to appease public fascination with alien life. But then came the signal. Samples retrieved. Return initiated. When Janus, 9 re-entered Earth's orbit, the welcome was restrained, almost sterile. A small capsule, no larger than a coffin, was parachuted into the Nevada biocontainment zone under the cover of night. No fanfare, no press release, just steel briefings and security clearances. Inside that capsule, kept at sub-zero and wrapped in thick carbon insulation, was a canister of ice and sediment unlike any ever seen. The first material ever brought back from Europa's alien sea. The facility that received it had no name, only a code name: Chimera Bay. Built underground, its corridors hummed with refrigeration coils and low-frequency static to muffle electromagnetic feedback. Within its lab, cleaner than any surgical theater, 6 scientists hovered over the canister like ancient priests about to open a relic. Dr. Ashira Vale, molecular biologist and exogenome specialist, held the honor of first contact. Her breath fogged behind her face shield as she uttered, "Begin observation protocol." At first, the samples seemed inert. Frozen sludge and microcrystalline debris suspended in saline, almost poetic in their dullness. The microscope revealed fibrous structures, vaguely cellular, but none matched known taxonomy. They moved, but only slightly, like sleeping organisms breathing once per century. The team logged it as microbial stasis. But then the readings began to change. Slowly, subtly. On day 3, a single strand of RNA emerged from the slurry. Not one, but many, like threads pushed through a needle in reverse. It didn't replicate like Earth life. It unfurled. The data logs showed base sequences that reassembled themselves depending on who was in the room. Not just chemically reactive, observationally responsive. Ashira noticed it first when she reviewed the footage. Every time her reflection crossed the biosample glass, the RNA curled tighter, as if flinching. Other times, it pulsed faintly, mimicking the rhythm of her footsteps. She dismissed it as coincidence until she isolated the lab feed and witnessed it contract in sync with her voice. She didn't report that. On day 5, technician Deeks noticed a strange flicker in the lab's environmental chamber. Infrared footage showed warmth rising from the sample, impossible under the controlled temperature. When they applied ultraviolet light to examine the reaction, the RNA rearranged itself into a spiral that almost looked architectural, like a blueprint. When Ashira slept, she dreamed of it, spirals in stone, helixes spiraling into the sky like cathedral towers. She awoke with its shape still spinning behind her eyelids. By day 7, the sample had tripled in volume. Not grown, just reorganized. The team tried to explain it through sublimation or internal gas pressure, but no chemical reaction could account for the way the ice inside the container never melted, yet the volume expanded. It was like the sample was adding something to itself from nothing. The night of day 8, the lab's AI system pinged a warning. Unscheduled vocal resonance detected in level 3. No human was logged as present. When security played back the audio recording, they heard what sounded like multiple voices, low, fragmented, and overlapping like a broken chorus. One phrase repeated, "Observer acknowledged." Ashira didn't sleep after that. She stayed late in the lab that night, eyes bloodshot, reviewing the spectrometry logs. She watched the data crawl across the screen like veins beneath translucent skin. The sample was evolving, responding. Not in hours, in minutes. Every new test, every touch, every glance seemed to provoke it into reconfiguration, like it was learning or remembering. And in the silence of that sterile lab, surrounded by layers of steel and vacuum seals, Ashira whispered to herself the thought she had refused to speak aloud until now. It knows we're here. Outside, far above the Earth's surface, satellites drifted quietly through the black. But several of them had begun to fail, sensors fried, optical feeds turned inward. One, in geosynchronous orbit, showed a brief flash of unknown interference before it too went dark. Its last image? A live feed of the Chimera Bay facility, zoomed from above, its coordinates never publicized. A coincidence? Or a warning? Back in the lab, the sample quivered slightly under the microscope, as if breathing again. They began calling it the window, the brief phase each day when the sample inside the canister changed. It always occurred between 2:04 and 2:13 a.m., no matter the human schedule. The lab's lights dimmed slightly during that time, though no one ever adjusted the brightness. Clocks drifted off by exactly 3 seconds and corrected themselves without syncing. Devices rebooted without cause. The sensors monitoring the sample recorded electromagnetic interference, not seen since the early Cold War, like a scrambled satellite transmission from something not quite digital.
Ashira didn't speak much that week.
She kept a private notebook, something with paper and ink, something offline. Inside it, she drew sketches of the structures forming in the ice. Tall, spindled towers like alien coral, domes spiraled in fractal patterns, knots that seemed to unfold mid-sketch into shapes she could not explain. She told herself it was stress-induced pareidolia, but in the corner of every drawing, without conscious effort, she kept writing the same phrase, "It's building itself." By day 9, the sample filled its containment chamber by 42%. Still sub-zero, still sealed. But inside, it was no longer just strands and crystal. There were tissues,
Veins.
pulsing membranes that contracted in timed intervals, like lungs that didn't need air. Dr. Costas called it an advanced protein bloom. Dr. Lee disagreed. It was too coordinated. Not decay, not growth. Assembly. The term mutation no longer applied in the genetic sense. This wasn't evolution. It was intentional transformation, as though guided by blueprints encoded not in DNA, but in behavior, ours. Cameras captured the clearest evidence when Ashira reached into the external sensor glove on day 10. Her movement caused the organ-like structure inside to flatten, then briefly emit light through its thin membrane. Bioluminescent, responding directly to her proximity. That night, when she reviewed the footage, she noticed the light pulsing not randomly but in rhythm. Morse code. It spelled a single word: begin. The lab team didn't believe her. The system showed no Morse pattern, but Ashira was certain. She had memorized the pulses by then, seen them behind her closed eyes, playing like a metronome at the edge of sleep. Later that day, Deeks collapsed in the hallway. Internal bleeding. No visible trauma. In his hospital scan, doctors discovered something lodged in his bloodstream, tiny black fragments, smooth and symmetrical, like artificial pollen. No one could explain how they entered his body. They matched no known element on Earth. Then came the second failure. Chimera Bay's air filtration system began misfiring. The building's oxygen levels fluctuated. Alarms didn't sound. When Ashira checked the logs, they had been erased and rewritten with new code that wasn't in any known programming language. That night, she stayed in the lab after everyone left. She sat beside the chamber and spoke out loud, not to the sample, but into the stillness, just to see if the anomaly would respond. She recited the Fibonacci sequence, the digits of pi, atomic weights, names of deep-sea organisms. Nothing changed, until she stopped, and then the pulse resumed.
Faster.
Matching her breathing. She pressed a finger to the glass and whispered, "What do you want?" The lights flickered. Not out.
Not dim.
Just different. A tone filled the room, like a distant wind through a canyon, but stretched through modems and dreams. It wasn't loud, but she felt it in her teeth, behind her eyes. A tone that didn't come from outside, but from inside the lab itself. The sample didn't glow this time. It throbbed. Rhythmic. Deliberate. The next morning, the structure inside the sample had grown fingers. Fingers. Small, translucent appendages curling around internal tendrils as if trying to grasp something just outside the glass. Each had 3 joints and lacked nails, skin, or bone. But they moved, not spasms. Intent. Dr. Costas suggested shutting the project down. Too much unpredictability. But Ashira argued against it, though she wasn't sure why. Part of her felt compelled, as if stopping now would be more dangerous than continuing. By day 12, the team confirmed that certain parts of the sample mirrored biological structures from Earth organisms. Sea cucumbers, starfish, fetal tissue, retinal cones, nerve bundles, as if the sample was constructing a body out of memories pulled from life itself. Not its own memories.
Ours.
At exactly 2:04 a.m., the window opened again. Only this time, the pulse didn't emerge from the sample, but from the building itself. The containment chamber's titanium frame trembled. Electromagnetic readers flatlined. The lab's AI shut itself down. For those 9 minutes, no one remembers what happened. Surveillance footage from every camera in the facility froze, just static. All biometric records from the team during that time were unreadable, showing impossible data. Heart rates of 300 BPM, skin temperatures below freezing, blood oxygen over 100%. When consciousness resumed, the sample had changed. There was something now curled at the center of the chamber. A mass of tissue, coiled like an embryo, half-formed, but undeniably humanoid. And written in condensation on the inside of the viewing glass, in perfect English. "You were always the sample." Ashira didn't sleep after the words appeared on the glass. She stood there for hours, trying not to breathe too loudly. The embryo inside the containment unit didn't move. It pulsed slowly, like a second heart in the room, but it didn't shift, didn't reach, didn't open its eyes, though somehow she felt they were open. Not toward her, but through her. By morning, the words had evaporated. The team assumed she imagined them, hallucinated under stress. But Ashira knew better. The humidity sensors confirmed moisture levels had risen unnaturally between 2 o'clock and 2:10 a.m., the same 9-minute window. Condensation was not a hallucination. The sample had stopped growing, at least visibly. But the anomaly wasn't done. That afternoon, another team member, Dr. Lee, reported a strange echo in her headset while working on unrelated tissue analysis. Not noise. A voice. Her own voice, whispering in reverse. "It's waking up." They ran a scan of the local network. No interference. No known signal input. But Lee wasn't alone. Soon, across Chimera Bay, others began hearing faint imitations of themselves through speakers, in security footage, even in phone voicemails left days earlier. The phenomenon wasn't contained. One of the biology interns brought in a petri dish from the genetics wing, a sealed sample he had left untouched since day one. It now contained a film of the same translucent tissue found in the Europa canister. He hadn't exposed it. He hadn't even approached the chamber. They tested surfaces, gloves, vents. All clean. Still, more samples began appearing in other labs, on glass slides, beneath microscopes, even inside closed autoclaves. Somehow, it was duplicating itself. Not by spreading, but by remembering where it had been. Ashira coined the phrase, memory-based replication. It wasn't viral, bacterial, or chemical. The organism remembered its interactions, then reinstated itself at points of prior contact, like echoes from a dream replayed in the waking world. On day 15, something appeared in Ashira's home. She lived alone, 3 miles from the facility. That morning, her bathroom mirror fogged without steam. Across it, letters etched themselves with unnatural symmetry. "Where's home?" The handwriting matched her own. She returned to the lab that evening and ran another simulation. This time, she tried to speak to it not as a scientist, but as a being. She wrote messages and left them on the chamber glass. Questions like, "What are you?" or "Why Earth?" or "Do you remember Europa?" Each time, no response. Until she asked, "Are you from here?" The lights in the room dimmed. The embryo twitched. And then, a single letter appeared across the lab's primary data terminal. Why? Not yes. Not Hugh. Just why? The replication spread again on day 16, this time into non-biological matter. Lab notebooks began appearing with pages duplicated in different handwriting. Pens scribbled without touch. In one storage room, glass petri dishes stacked themselves into spiral formations. No one witnessed it. Cameras didn't show movement. But every dish recorded weight changes, like something had entered and exited each one, silently. Dr. Costas quit the project that night. He left without resignation, leaving his keycard on the floor beside the elevator. Later, Ashira checked the building's internal sensors. Between 3:12 and 3:15 a.m., his biometric scan appeared twice. Once heading out as expected, but a second time 13 seconds later, walking back in. Only no one ever saw him again. Security footage was blank. The anomaly was no longer isolated to the sample. Ashira realized the organism or intelligence or whatever it was rewriting cause and effect. Replication wasn't limited to space. It was now altering sequence. Something that had left could reappear. Something that had died could return. But not exactly as before. The differences were subtle. Posture, tone, gaze. As though reality itself was beginning to glitch around the presence of this thing. Then came the message from Europa. A transmission. Static at first, but filtered through low-band radio, a clear signal emerged. One word repeated over and over, distorted and looped. Host. Not a greeting. Not a threat. A designation. The Europa organism wasn't the entity. It was the vessel. The entity had come with it, and it was no longer confined. Ashira checked her blood that evening. A personal test unsanctioned. What she found chilled her. Low concentrations of the same unidentified proteins found in Deeks. No symptoms. No pain. But when she tried to sequence it, her own DNA interrupted the process. A recursive loop formed in the sequencing algorithm. It wouldn't stop calculating. Her genome had been edited. Quietly. Invisibly. She remembered the embryo.
The twitch.
The word. You were always the sample. Not a metaphor. Not fear. Instruction. They had thought they retrieved something from Europa. But perhaps Europa was just the return address. By day 18, Chimera Bay was no longer a lab. It had become a listening post. Not for signals from deep space, but for whispers already here. The Europa organism, the presence was no longer confined to samples, dishes, or glass. It had moved into behaviors, memories, and time itself. Surveillance logs didn't match reality. Employees found fragments of conversations they hadn't yet had, notes written in their own hand with dates from tomorrow, and dreams that bled into waking hours like spilled ink across a white page. Ashira began cataloging the disruptions as temporal intrusions. But the term felt inadequate. It wasn't just time folding in on itself, it was being rewritten. A decision had to be made. The breach protocol was always theoretical, a last resort set in place for pathogen-level threats. It required 2 directors' biometric approval, and even then, couldn't be executed without oversight from Homeland Biological Defense. But Chimera Bay never made that call, because the system was already overridden. At 2:44 a.m., on day 19, all doors sealed simultaneously. Emergency power routed itself into the Europa chamber. The lights dimmed. The embryo, silent for days, began to vibrate. Not visibly, but instrumentally. It began broadcasting signals into the facility's electrical grid, pulsing in binary waves at a frequency slightly above human hearing. Everyone stopped moving. Not from fear. From knowing. They felt it. Inside them. The signal wasn't an alert. It was a countdown. In her office, Ashira opened the last file in her private archive, an auto-recording from the first Europa extraction drone. It had never been fully decoded. Too much corruption, too much feedback. But this time, when she ran it, it played clean. Not visuals, just sound. It was her voice. A perfect copy, but younger, more certain.
Saying,
the breach begins with knowing. The breach ends with forgetting. And then silence. the kind that feels old. That was when the stars changed. Outside the lab, night fell fast, too fast. Shadows bent in unfamiliar angles. The moon was missing. The sky above Chimera Bay shimmered like film submerged in fluid. When Ashira looked up from the rooftop, she didn't see stars.
She saw eyes.
Not blinking. Not human. Just watching. Like the observatory was now the observed. Inside, the embryo began to liquefy. Not dissolve, transform. Its structure fractured into smaller, self-similar nodes, each spinning with impossible motion, like tiny galaxies collapsing and forming at once. It was no longer just biology. It was instruction code. Alive, yes, but informational. It didn't want to live. It wanted to rewrite. Ashira realized too late that Chimera Bay wasn't a lab built to study Europa. It was a lab built by the thing from Europa. The entire facility was part of the signal path, a transmitter constructed by human hands following imperceptible blueprints hidden in curiosity, ambition, and false discovery. The stars hadn't moved. They had waited. She tried to issue a local EMP burst, but her access was gone. The lab's own systems had rewritten their command hierarchy. Every screen across the base displayed the same word, again and again. Seeding.
And then,
Begin. Outside, the sea began to boil. It didn't rise. It didn't crash. It simply heated, quietly, deliberately. Sea life along the coastline surfaced, belly up, dead. But the water glowed. From beneath the surface, structures began to bloom, metallic roots coiling upward like reversed lightning bolts. A second lab, growing beneath the ocean. Replication again, but on a planetary scale. Ashira's last act was to walk into the cryogenics wing and manually inject a blackout agent into her bloodstream, a mixture designed to scramble all bioelectrical signals and deny access to her cognitive pathways. It wouldn't kill her, but it would silence her mind. She laid down in the isolation pod, sealed the hatch, and entered a blackout dream. No thoughts, no motion, just quiet, until the first whisper returned. Not her own, not from Earth, from beneath Earth. A childlike voice, layered with radio static, echoed in the dark. You are not the host anymore. The pod sealed. Chimera Bay was no more. But across the globe, unrelated observatories began detecting anomalies, flickers in star maps, voices embedded in solar radiation, dreams aligning across unconnected minds. One astronomer in Chile awoke to find a map carved into frost on her observatory window. It didn't match any known constellation, but it matched her childhood drawing of the night sky. One she had never shown anyone. One she had drawn before Europa was even discovered. And in the center of the drawing, a pulsing dot, labeled in her own handwriting, Ashira.